Welcome to the April edition of BBRO BeetCast. I'm Francesca Broom, your host and Knowledge Exchange Manager for BBRO. We've had a great start to the season with drilling well underway. And with aphids due in May, it's been vitally important to drill early and in the right conditions. With a little bit of moisture and the warm soils, the crop will soon be away. We hope that some of you have made use of our soil temperature data from our weather stations, which is available on the BBRO website. We have found it invaluable and we hope you have too. I always find this time of year quite exciting when you're waiting for those first few green shoots to appear, and even better if they appear in a nice straight, even drill. But what if the crop struggles to establish? Could you have a pathogen lurking beneath the surface of your soil? Dr. Suzannah Harder joins me now to discuss her new work investigating seedling diseases. We've got a sunny day outside and I know most people are out with the drills, and we've started to put the sugar beet seed in the ground. And now is the time to think about what might come in the next few weeks, isn't it? I'm joined here today by Dr. Suzannah Harder, who's our newest recruit at BBRO, but certainly not new to BBRO in her career, because she was with us first of all as a young student and then as a PhD student for 4 years.
Yes, 4 years.
Yeah, so welcome to BBRO, your first proper BeetCast, I think, isn't it?
Yes, I'm a newbie to this.
So welcome along, and I promise we won't go too hard on you.
Thank you.
But you have a vast experience in sugar beet, and particularly on virus yellows and your work you've been doing in the past. So do you want to just give us a little bit of an overview of your PhD study?
Yeah, so that was started in 2020, all about strain variation in virus yellows. So the idea of looking at whether, when we focus on beet mild, for example, is there different strains of it, maybe one that's particularly nasty or not. As it turned out, the biggest finding was actually that our in-house beet mild culture was a little bit weak, and explained some of the results Alistair had been finding in Goliath. So we've changed up our cultures here to make sure they're staying representative of what people are finding out on farm.
And some of the growers that attended our previous Beet Tech events may remember you did a really brilliant presentation on breeds of dogs against the varieties, didn't you?
Yes, I do like an analogy. Yes, it was if we had the Rottweiler equivalent, or if we were dealing with a little Chihuahua.
And now, obviously, full-time at BBRO, and you're looking in the sort of resistance and fungal disease side now, isn't it? But on seedling diseases.
Yeah, so I've not really dealt with fungal diseases before, Chaz. It's always been viruses, but I've taken a little bit of a step into the fungal disease world, in particular looking at seedlings.
And you're looking at different strains of fungal disease. So
exactly.
we're looking, when we had issues in the field previously with young seeds, it was blackleg, wasn't it?
Yes, so we tend to use that broad term blackleg to describe any instance where the hypocotyl, where that root stem area of a young seedling turns black and the seedling starts wilting. We tend to just use this general disease term blackleg for it. Or sometimes dampening off is also used as well.
Yeah, and that's been the case for many a year, but you're actually looking a little bit deeper, right, to see if we can find exactly what's causing it.
Yeah, so I think it's easy to think of a bit like virus yellows. You know, we use the virus yellows term, but actually it's different viruses that can cause it. It's exactly the same here as well. So we say dampening off or blackleg, but actually there are multiple different fungal pathogens that can cause the disease. And that's not really something we've looked at before, because they're really hard to diagnose just by looking at them. It tends to be very, very similar symptoms, and you need the molecular tools or fungal culturing tools to be able to differentiate between them. And to be honest, up until now it hasn't really mattered what's causing it, because our treatments on our seed has generally protected against it.
Yeah, and do you think the sort of climate change has had a difference to that?
Yeah, definitely. I think we're all noticing on occasion these warmer, wetter springs, and these diseases tend to really like those conditions, and that disease pressure in some fields is ramping up a little bit.
And are you looking for existing diseases that we know of, or do you think there's something else at play?
So generally I hope it's what we already know. So I think traditionally we've thought of Aphanomyces and maybe Pythium or Phoma, but they're sort of in our normal toolbox that we're aware of. Rhizoctonia we know about and is occasionally found, but we've generally considered it needs a lot warmer soils, and so isn't really considered a widespread problem in the UK. But there's been some American research in recent years showing that actually it can grow and germinate at lower temperatures, and that combined with our perhaps warmer temperatures with climate change means it is a little bit more on our radar and something we need to try and monitor and look out for.
And in previous years, when would we have been able to identify Rhizoctonia as being an issue?
Well, we wouldn't really, and that's the thing. So we would just see blackleg symptoms. So the only exception to the sort of symptom rule is Aphanomyces. So Aphanomyces infection, you'll see black leg of the stem or the root going black, and then the leaves won't wilt.
Right.
That's quite a unique symptom. All the other diseases cause the root blackening, stem blackening, but then the leaves die and wilt too.
So tell us a little bit more about your work. So you're actually going to be investigating, I take it there's spores within the soil?
Sort of, yeah. So the added complication is, although we've been talking about fungal diseases, some of the diseases are fungal, some of them are actually called oomocytes.
Oomocytes, okay.
Yes.
so a new word for the sugar beet dictionary is this one?
Yes, so traditionally we would have called them all the same under that sort of broad fungi umbrella, but now with sort of newer classifications, things like your Aphanomyces and Pythium are actually oomocytes. They're very similar, but they're water-loving, and they produce these, I say cool, but I think they are little spores, zoospores, that's another new word for you, and they have little tails and they'll swim through the water, which is a little bit different to the other fungal diseases.
Okay.
So that's what's infecting your sugar beet roots, and we could target them, but they're quite fast moving and quite hard to capture. So at least to begin with, I'm aiming at doing DNA extractions on symptomatic plants, symptomatic seedlings, and then using some of the molecular techniques in the lab to drill down into identifying those little fragments of genetic code which will tell us which species is causing the problem.
And you sound quite excited about this. I mean, I'm not sure whether the growers would be, but we do need to drill down and actually find out what the problems are there. But, you're talking about tiny little spores, and yet you make it sound like a tadpole. So in size, what are we looking at?
Size-wise, you're looking at needing sort of down the microscope, we're at sort of a 10,000 times magnification to be able to spot these things. We are definitely on the microscopic here. Though saying that, once they've infected, they are a bit like, some of the others are a bit more like a mushroom, and you get this sort of mycelial growth that you might notice different species, but say when you open a compost bag and you get that sort of white mycelial growth, it's a similar thing there. They do get to the size that you can see, but no, the spores are tiny. Yeah,
so this is not something that growers will be able to detect themselves, is it?
No, we are very much lab-based here.
Yeah. So in that case, if a grower has a case of blackleg and they want to know what's behind it, what will they be able to do in future?
So at the moment, if you were to send that into plant clinic, unless it was that classic case where you've got a blackened stem but unwilted leaves, in which case if it's Aphanomyces, we would just say blackleg and we wouldn't be able to really diagnose any further. But the plan is to develop these diagnostics, and in future we would be able to tell you whether it was Aphanomyces, Rhizoctonia, Pythium or even Phoma.
Okay, so once that's been diagnosed and you can do that, what's the next step? I mean, is there different treatments?
So things are coming, and they are certainly on the horizon. I think traditionally we've been very reliant in recent years on tachygyrin, but that is changing, and I think in the future there's going to be a lot more choice for growers, and hopefully having a little bit more detailed information will aid that decision of which treatments to use and when.
And I'm assuming these treatments are going to be seed treatments, aren't they? So
yes,
people will need to know that they've got that disease in the ground before they actually agree to whatever seed treatment they're going for that year.
Yeah, I guess that will depend on sort of the authorisation and regulation, whether you can use it prophylactically. For example, in the case of tachygyrum, we don't get much protection against rhizoctonia. So if you knew you had a rhizoctonia problem, you might opt to use something in addition to as well.
So once a grower is aware that they have an issue within their soil, obviously they'll be able to go for a seed treatment. And is this going to be a little bit like with a BCN, that once you know you've got a cyst in the ground, that you are going to have to continue to treat your seed accordingly, or you never know, a variety might come on board, but the problem's not going to go away?
No, yeah, exactly. So these diseases are really persistent in the soil, and you would then look to use either the seed treatment, or you're right, then on the continent you do get varieties showing resistance to some of these diseases, which in the future we might find a need for here as well. But it would be a case of, if you knew you had the disease, to then make your choices accordingly for the foreseeable future.
Mm-hmm. Having gone from investigating virus and doing your research on that side into fungal diseases, any preference?
Yeah, I'm afraid I'm still a virologist at heart. Fungi and oomycetes, there's so much, I appreciate you can see them, which some people might think makes it easier, but they've got way more complicated life cycles. They're not as neat as my virus friends.
Virus friends.
Yeah, that doesn't make me sound very cool, does it?
Yeah, I was going to say, I don't think there are many growers that would agree with that one somehow. But
sorry.
So obviously your work's still early days, really, isn't it, what you're investigating? But do you need the support of growers in order to push this research further?
Yes, Jez. So I think we're very much in the early days of this project this season, but as we go forward, I will need the support, particularly with any disease samples people might notice in their own fields. I guess if any growers had a really severe problem this year, it would be really interesting to get some samples through the plant clinic or contacting me directly to understand a little bit more about what's going on there. And potentially next year there might be a plea going out for any symptomatic samples to be sent in. Yeah,
because at the moment you are just refining the diagnostics, aren't you? So it's going to be getting those right before you can really do testing and give a definitive answer to anyone.
Yeah, definitely. And you say defining, but in some cases there aren't tests easily available. So this is really back to the lab bench and trying to get the fundamentals right before we start rolling this out too much.
I hate to say it, Suzannah, you might prefer the viruses, but it looks like you're creating a little niche for you here at the moment, looking at fungal diseases, and that might be where we have to put you for a little while as yet.
Yeah, I mean the project is for 3.5 years, so it's going to take a little while to get this one sorted, shall we say.
Yeah, well, and I'm sure not only will you be pleased to probably finish it, but the growers will certainly be appreciative of the work that you've done and actually making the whole of the sugar beet industry healthier. So
hopefully.
Thank you very much.
Thank you.
If you attended one of our BeetTech events, you will have heard Suzannah talking about a plant clinic case on rhizomania. It was unravelling that case that led her to her current work on soil-based pathogens. Therefore, we can't really finish without hearing the outcome of her earlier work. And we return to the stage at BeetTech 25.
This is just to point out that we don't tend to have an average year anymore. I think we all knew that, but springs are perhaps getting a little warmer and a little wetter, and that is changing our disease pressures as well. We've had a few examples of this that we could perhaps pick out. Beet moth, that was something that I don't think we'd ever really seen. It came in quite suddenly. I particularly noticed it in 2022. That was the first year when I was doing my PhD. I ventured out into the field for the first time, left the safety of the lab. Field trial was completely decimated by those particular moths, much to my dismay. Another example, Cercospora. When I first started at Bee Bureau, if we had an example of Cercospora into the plant clinic, it was really quite exciting. It was quite a novelty, whereas now we've got whole Bee Bureau projects dedicated and Spornado profile right across the country trying to monitor its arrival. But it's not just new diseases that are changing. with climate change. We've also got some things that perhaps we are familiar with but are being more favoured by these conditions. And it's for that reason I want to talk about revisiting rhizomania over the next few moments. To do that, I'm going to use a real plant clinic case. We're just going to go through and hopefully show you the importance of detailed diagnostics in these times of sort of changing disease pressures. Plant clinic case came into the lab. We'd got yellow leaves, really fluorescent in colour. I'm not talking the orangey colour that you might see with virus yellows. These were really fluorescent yellow, quite different. Lots of lateral roots, but no cysts. This wasn't BCN, and the roots had a quite unique shape, almost wine glass shape, constricted. This case, we did have some really good pictures, some really good information, and that enabled us to start thinking rhizomania. What is rhizomania? It's a virus. So we're beet necrotic yellow vein virus. It's an RNA virus. We'll come back to that in a moment. And it's transmitted by a parasitic protist, something that we might once have called a fungi. That's not quite true anymore, called Polymyxobetae. And it's that organism that's favoured by the warm, wet conditions. Those warm, wet soils will enable it to infect your roots. And if it's carrying the virus, you then get your rhizomania infection. The Polymyxobetae lives in the soil, like I say, and can survive for very long periods of time as resting spores, decades. So once a field is infected, you have rhizomania in that field virtually for perpetuity. I am very conscious in the audience that those of you who were farming in the 80s are all too aware of rhizomania. But, and apologies to Mark, who I know worked on this in his early career, that was a good 30 years ago. So for the benefit of youngsters in the audience, let's have a little recap of our timeline. So rhizomania was first identified in Italy back in 1952, and it was really starting to cause some major problems. It spread consistently across the continent and further afield, and we were watching it as an industry, growing increasingly concerned, and tried our best to stop it reaching our shores. So we had legislation in place, things like limiting imports and exports of sugar beet roots and soil. But ultimately, it did make it to Suffolk in 1987. But thankfully, breeding efforts, and like Mark was saying earlier with SBR, knowledge of the continent meant that we had our first rhizomania variety introduced in 1997. As an aside, that was also the first year it was reported in the Newark factory area. Variety adoption was really quite successful, and by 2013, the entire UK crop was sown with partially resistant rhizomania varieties. And the disease had been declared endemic at that point, and your legislation controls dropped. I say that the entire crop was sown with partially resistant varieties, so much so that now it's almost hiding in plain sight up there in the top left of your recommended list. It almost goes unnoticed that all your varieties are RZ1. But like I said earlier, we're dealing here with an RNA virus, and viruses do like to mutate. They like to change and evade us, and that is exactly what happened. The story's not quite so simple, because in 2007, we had some examples of disease symptoms showing in crops that were RZ1 genotypes. A series of genetic testing led to the identification of the AYPR strain of rhizomania. If we're really drilling down into the detail, AYPR, that comes about due to the really tiny changes in the RNA virus's genetic code. RNA, third fragment of the virus, changes to an alanine, tyrosine, proline, and arginine amino acid protein building block, and it is that tiny change which allows it to evade the RZ1 resistance gene. I admit that's possibly a detail too far, but that change has a big impact. Fortunately, the additional resistance gene, RZ2, had been identified, and combining those two resistance genes in the sugar beet variety meant that we could keep crops protected even from an AYPR strain of the virus. So back to our plant clinic case. From the information we were given, we knew it was an RZ1 variety, but we're still seeing symptoms. That really gave us in the lab sort of two options. Option one, this is a case of AYPR, but we didn't know was in the field. Or option two, this is something new and a bit scary. Another option, another strain has developed that we don't know about.
And that's where I come into the story. So I finished my PhD back in February last year. And one of my first tasks was to develop the Rhizomania diagnostics in-house at BBRO, make sure that we are able to identify these strains and in particular identify any novel diseases coming in. Traditionally, we've used an antibody-based method. That's a really good technique. ELISA is superb for getting through a lot of samples. It's very high throughput and can tell us whether the virus BMYVV is present or absent. But it can't do strain identification. For that, I had to bring in a slightly different approach. This isn't new equipment, it's not new chemistry, but PCR and gel electrophoresis is really specific. We can really drill down into the exact strains of the virus present. Not high throughput. I can't do a whole field scale analysis, but that's not really what this is about. This is getting absolute detailed diagnostics to identify if we have a serious problem or something that we can manage with varieties. And that's exactly what we did in this case. So by testing, using that new technology, we were able to identify that in this particular plant clinic case, we had the AYPR strain of Rhizomania present in the field that we previously didn't know about. And in a way, this is a good news story because we could suggest that in the future, we use an RZ1 and 2 variety in this field and hopefully limit the problem. Wasn't anything new or scary.
And on that positive note from Suzannah, we'll leave you for this month. We hope that drilling continues at pace and that by Easter we can all see those green stripes of success. Thank you very much for listening.